So I hope everyone can hear me well. So just let me know if you cannot hear me well, or if there is some interruptions and you can't clearly hear me. But let's get started. Oh, I need to share my presentation. Hang on for a second. And thing. So I have to activate description. So I have also to share the sound here. Yeah. I think it should be good. Let me check again. I think it should be good. Yeah. So today I'm going to discuss applications of Omic's technology to human nutrition optimizations. So it's definitely a pleasure or a joy for me to share one of the recent advances in life sciences with all of you. So only a few years ago, advances in engineering and computing technology allowed us to begin using multi-omics methodologies in life sciences and medicine in our research. So multi-omics, as you notice, this new, probably new term, new vocabulary, on the other hand, are a quite difficult concept to grasp. So in terms of the high learning curve to overcome, it might be comparable to understand climate change. So, but it falls under the category of a particularly challenging subject, such as system biology. This is due to the biological system intrinsic concatenation and orthogonality, for example. So I have to move to the next slide. Hang on. Yep. So our objective for today's lecture are meant to allow you, every one of you with a range of learning backgrounds, to first be on the same page, or at least have a similar knowledge of how life sciences work before learning what multi-omics is. For starter, you would be able to define what science is. What would be the scientific methodology? So there will be a clear distinction between science and non-science. The second goal is to gain a good overview of life sciences, or a snapshot of them. You'd be reminded of the biological phenomenon we are dealing with here. So we have a number of biological concepts, but today we will focus on one of the most basic, namely the concept of biological range. I hope you keep in mind that this approach will play a part in the design of multi-omics investigations. Third, you'll be able to describe multi-omics in the context of life sciences and give instances of some of the most commonly investigated omics in the recent years. The fourth task requires you to describe a multi-omics strategy to life science comprehension. To begin, what makes using multi-omics to solve biological, medicinal, or nutritional concerns so difficult? However, for our increasingly linked scientific fields, it is critical to maximize the use of multi-omics technologies. And finally, we'll talk about a multi-omics discovery that helps us comprehend the link between nutrient metabolisms and health. A recent publication with great multi-omics data relevant to nutrition sciences will be discussed at the end of the session. So now let's see what the scientific boundary is, so that it may be distinguished from non-science. So let us use the Science Council's definition of science. It's on the sciencecouncil.org website. The Science Council claims that science is the pursuit and application of knowledge and understanding of the natural and social world following a systematic methodology based on evidence. The crucial word here is evidence-based scientific techniques, methodologies. So what is exactly scientific methods? It consists of the following: objective observation, measurement, and data. So possibly, although not necessarily using mathematics as a tool for the measurement and to generate data. And the second one would be the evidence itself. And the third one, experiment and or observation as benchmarks for testing hypotheses. Then induction, meaning reasoning to establish general rules or conclusion drawn from facts or examples. Then obviously, we need a repetitions or reproducibility. And after all, we need a critical analysis of all the observed data, and then verification and testing. So this is critical exposure to scrutiny by using peer review and assessment. Life sciences and medicine operate within the boundary of this criteria of scientific methodology. So now, so far we've discussed what science is and how scientific methodology works. Science is how we learn about life. Natural sciences, such as the mind-bogglingly enormous universe, include life as a subset. There is only one planet, Earth, that contains life out of 10 to the power of 24 planets, or a trillion planets in the universe. So far, there are 59 confirmed habitable exoplanets orbiting other stars besides suns at distances that are literally out of reach, have been discovered after decades of research. Thus, there is an exceedingly small likelihood of life in the universe, with 59 out of the 10 to the power of 24 planets. So it also means that life exists and operates in a very, very small area of the universe. The range is also constrained by the Earth itself. So now the living phenomena that have been decoded through scientific methodologies are astounding. We now have a better understanding of different life concepts, such as the concept of biological range, the concept of biological nonlinear dynamics, biological randomness, biological concatenation and orthogonality, biological pause, biological immortality, biological threshold, biological nonlinear self-organization, biological communication, and speech. So as I previously stated, we will briefly cover one of the most basic concepts among them, namely the concept of biological range. So range here is defined as the area of variation between upper and lower limits on a particular scale. So we've got to think that living things or life mechanisms operate within a range. So let's grasp one of them and apply it, and then see along the way how you can think of this range in the study or in the concept or the presented concepts. So consider this biological range of visible light. The term light refers to a type of energy. Visible light has wavelength ranging from 380 nanometer to 780 nanometer, and is a portion of the electromagnetic spectrum. Our visual system is energized by light, which is reflected from things into our eyes and allows us to see. Thus, the electromagnetic spectrum has just about 400 nanometers of 10 to the power of 15 nanometers. So it's unbelievable. It's really small, like 0.0000 and so on, 4%. So here you can see it here on the left, lower right, the visible spectrum for humans versus bees. Here you can see it here on the lower right. So while the range of visible light for life is extremely limited, it is incredibly beautiful. So let us take the information about visible light to another level. While it seems the range of visible lights in nanometers is linear, life can process it differently based on environmental inputs. Here we have two populations or communities of life, namely humans and birds. While most of the light spectrum between humans and birds overlap, however, in the ultraviolet range, we can only see black, such as this blackbird, while other birds can enjoy the colorful fluorescences of the object. In this case, of the bird, fluorescence. This is the fur of the bird. So birds can see fluorescences in the object in the fur of this bird. So now to add a layer of complexity that affects the processing output is the latitude or height. The latitude here or height and time of the year when visual input is registered, as it is seen here in the graph depicting differences in registration of visual input between sea level and top of the atmosphere. Well, this is an easy example. So we are starting with easy. Hopefully, it's easy to grasp. And by now, you started to get a perspective of how the concept of biological range is a fact of life that happens at different levels of life. It is constantly active as long as life itself. The question is how to correlate different levels of different populations to obtain an understanding of whether what is global mechanisms that regulate such levels, and two, what particular molecules may govern such layers of a biological system. Let's get into more challenging yet exciting currently available technologies for scrutinizing those layers of life, which is the multi-omics approaches. So multi-omics are variously called pan-omics or integromics, integrated omics, polyomics, transomic or cross-omics. So you came across those omics name, literally it's multi-omics. We use multi-omics from now on. It aims to combine two or more omics data sets to aid in data analysis, visualization, and interpretation to determine the mechanisms of a biological process. The term omics stems from the suffix om, added to many fields of biological study, which refers to the study of something in its entirety. There are estimated to be over 500 omic types. As you can see here in the list of variety of oms and omics you may never heard before. And some oms or omics are quite strange, definitely. So anyway, I'm not going to go into the list, but people discover, and then you can even discover name omics as long as you are studying of something in totality, in overall or its entirety. So in life sciences, multi-omics is an approach aiming to improve the understanding of system regulatory biology, molecular central dogma, and genotype-phenotype relationship by combining 3 or more different omics data. As you can see here on the right side, the current central dogma of life is the flow of genetic information within a biological system, from DNA to RNA transcription to protein translation, and obviously to metabolites, to enzyme catalysis. You can see here, I have blocked a DNA to RNA and protein. You cannot separate these three. So if DNA, if you're talking of DNA, the genetic expression, you cannot talk only DNA, but you have to talk about DNA, RNA, and the protein as the product of RNA. So this is 3-in-1 of genetic expressions. So it is no longer linear from DNA to RNA to protein, but there are interplays among DNA, RNA, protein, and of course the metabolites in directing genetic expression. So most common omics in life sciences, you will hear it from time to time. It's genomics, epigenomics, transcriptomics, proteomics, metabolomics, and phenomics. So phenomics is the study of observable traits here. So meaning there is a genetic underlying to be elucidated. For example, observable symptoms such as morphological differences, behavioral traits, developmental patterns. And then genomics, if we understand phenotypes, we want to get to know what is the level of genetic expression. We can even trying to understand on the DNA level itself as in totality. Or so the study of sequence level DNA modifications that would correlate to the phenotypes. For example, single nucleotide variation or base changes, substitution, insertion, and or deletions, structural variations such as copy number variants, or a chromosomal variation such as segment aneuploidy, whole genome, or whole chromosome alterations. And of course, there is what we call an epigenomics, the study of non-sequence level DNA modifications. Examples are DNA methylation here. RNA modifications such as methylation or phosphorylation, histone modifications such as methylations, acylation, phosphorylation. So, and all of this genomics, epigenomics would affect the transcriptomics depending on the time and the space, the situation, and on the different level of range. So it would yield different kind of transcript or genetic expression, and we can study it by transcriptomics. So this is the study of RNA transcript abundance and expression. For example, coding RNA transcript such as messenger RNA, non-coding RNA, transcripts such as circular RNA, microRNA, long non-coding RNA, transfer RNA, ribosomal RNA, small nuclear RNA, small nuclear RNA, small interfering RNA, numbers of species of RNA. And obviously the expression would need an understanding of the protein level. So proteomics, the study of protein abundance and expression. So the examples here are antibodies such as immunoglobulins, enzymes such as polymerases, messenger proteins such as growth hormones, structural proteins such as actin, and transport or storage protein such as ferritin. And protein would yield a small metabolites, byproducts, secondary metabolites. So the study of low molecular weight compounds in a cell, for example, thousands of metabolites have been documented in biological examples such as acetic acid and so on. So if you can imagine all of this, a few omics in life sciences that just comprising the core genetic expression at the level of genetic expression, you can imagine. So within this central dogma of life, you can imagine this is massive. It generates, if you've studied this, you can imagine it would generate massive data. So we'll find out multi-omics approaches in life sciences by answering the questions, why is multi-omics challenging? So firstly, its individual omics analysis presents a multitude of challenges. Hence, multi-omics analysis inherits challenges from the single omics data sets and confounds further analysis with other new challenges of the integration fusions, clustering, visualization, and functional characterizations. For instance, prior to integrating 2 or more omics analysis, the investigator can face challenges in terms of data harmonization, meaning different data scaling, data normalization, and data transformation needs pertaining to individual omics data sets. And further, given dimensionality of constraints posed while integrating large multi-omics data sets, for example, in a large population study with thousands of individual samples, the computational burden and storage space requirements can be limiting for a given study. The second thing, what we call data wrangling, or we refer it as data munging. So includes various levels of transformation and mapping, and is critical to the multi-omics fields. So transformation is accomplished by data scaling, normalization, and imputations. Imputation versus the computation. So the imputations to fill the gaps where there is an empty data. Where there isn't any data. We need to impute in that data that helps harmonize different omics data together. So, and the category of mapping can be the process of harmonization of identifications across various omics data types, or simply annotating data across available metadata. A labor-intensive process that requires massive one-to-one or one-to-many relationship operations. So now, careful registration of samples and robust metadata recording tables with the involvement of data generation and analysis teams can help circumvent this challenge and mitigate errors. Next, data heterogeneity is often another bottleneck while dealing with multi-omics data, as these are generated by varied technology. So consider sequencing versus mass spectrometry, or microarray versus mass spectrometry. What else? And the platforms, targeted versus untargeted, high resolution versus single cell. So the pre-processing steps pertaining to individual data sets might not help overall, especially when democratizing them under a unified framework still remains challenging. However, some tools have led to improved handling, such as similarity network fusions or mixed omics, multi-omics factor analysis, MOFA, among others. These are the tools to attempt to network, to bridge, bridging the gaps, bridging between different platforms. And of course, data representations by means of dimensionality reduction that intends to project relationship of features such as SNPs, single nucleotide polymorphism, transcripts, proteins, metabolites across observation. Observation meaning samples, the conditions, different omics layer in a reduced space. So it's common practice a priori in multi-omics efforts. Now, typically following pre-processing after data normalization, data representation is applied to identify outliers, technical sources of variation such as batch effects, and obvious biological patterns at each level of analysis, such as feature identification, extraction, and selection. This exercise aids in learning biological patterns and relationship of the data in bias identification and mitigation via appreciation of technical factors contributing to the noise, adjusting them via batch effect correction, and identification of groups or subgroups to confirm hypotheses of phenotypic conditions of interest in a given study. So this is achieved by using clustering methods that are k-means density-based or graph-based, followed by generating visual representation using dimensionality reduction methods, such as principal component analysis, PCA, the distributed neighbor, the SNA, or uniform manifold approximation and projection UMAP. So this is some of the statistical vocabulary here to capture linear and non-linear difference relationship in the data. So, well, different omics data require different numbers of samples to draw reliable conclusions, and reliability is dependent on false discovery rate, FDR, which is influenced by the number of measured entities, such as transcript proteins or metabolites. Now here on this slide is an example of complexity and interconnectivity of omics data sources in a multi-omics framework. A simple cellular endogenous metabolite, lactate, is biosynthesized enzymatically from pyruphate, another metabolite, right? So here, metabolite, this is lactate. Lactate, it's coming from pyruphate here. With the help of lactate dehydrogenase. So this is a 3D structure of LDHA, lactate dehydrogenase, a catalytic protein. In turn, this LDHA can interact with several known and unknown proteins through protein-protein interactions to regulate its own function. And itself is subjected to diverse post-translational modifications. That this is post-transcriptional modification, PTMs, that regulate its catalytic function. So lactate measurement through techniques such as in vivo brain imaging in humans or other model animals can generate lactate spatial distributions. So this is an imaging. You can see the lactate spatial distributions in the image, in the image section of brains. So now the gut microbiome, via lactobacillus and other microbes can synthesize lactate and release it into human physiological system to contribute to lactate level. So now we jump to different situation here. When you consider microbiome, namely in the gut microbiome. So if lactobacillus can synthesize lactate and contribute to the physiological systems and contribute to the lactate levels. Lactate biosynthesis regulation can be due to various levels of genetics. So here is the genetic levels. For example, SNPs, single nucleotide polymorphisms, or copy number variants, and so on. Transcriptomic or post-transcriptomic, such as microRNA, or epigenetic, such as DNA methylation here. Changes on the LDH gene here. So though this is one of the well-studied sets of multi-omics interaction, one can expect more complex and unknown interaction while integrating multi-omics data set. Now, some of the considerations needed to overcome these issues include careful assessment of statistical power and effect size appropriate to the experimental design. Identification of confounders, such as sex, age, input materials inherent to the data, biases, such as replicates or biological or technical, both actually biological and technical during the replication, so that potentially gives you biases. And sources of variations, bets, analytical or unwanted, that are anticipated in the course of data generation. Quality assurance and quality control measures that are associated with individual omics, data generation and analytical platforms. And of course, cross-validation measures are implemented in cases of unavoidable biases. Now, multi-omics approaches can broadly be categorized as supervised, meaning classification tasks that include discrete outcomes, such as disease or control status, and prediction tasks like that of the continuous outcome, survival or pain score, for example, and exploratory, unsupervised clustering, so disease subtype discovery and relationship-based analysis, such as correlation or covariance and network models. So here's the list of tools available to run interactive multi-omics study, and we have what we call FAIR, findable, accessible, interoperable, reusable research data compliance. So these tools are FAIR compliance. So findable, accessible, interoperable, and reusable. So you will see it's the list is quite exhaustive. So popular emerging multi-omics tools such as Omics, and the purpose is a tool with a framework that provides wide range of multivariate statistical methods for exploratory data analysis. It involves features identification, extraction, and selection, and you can get, if it happens to match with your study, you can take a look, check it out at mixomic.org or with their publication. MOFA, I mentioned earlier, a probabilistic multi-omics factors analysis-based framework that involves EDA and data integration. This is super unsupervised. SNF, Myodin, Paintomics, Freeomics, and then there is a data sharing. For example, Omics DI, Zenodo is a general purpose open access data software repository that allows users to obtain a citable DOI. Zenodo.org, OSF, an open platform to enable collaboration by registering research project, materials, data, and documentation, and so on. So code sharing. So we are familiar, some of you are familiar with GitHub, a version control code sharing and collaborative platform. You can find different kind of source code at github.com, and whatever the purpose, and you have to find out which one that would match to your purpose. And then there is a workflow sharing, environment sharing. Some of you are familiar using Conda or BioConda, Docker, biocontainers, and so on. And data visualization. For example, Shiny, a framework in R for doing GUI-based interactive applications. Plotty, a cross-language interactive plot library. Or Cytoscape, a platform for network data integration, analysis, and visualization. You can now you can imagine all the computer scientists, bioinformaticians are quite active, productive, attempting to develop multi-omic platform. Trying to harmonize between a different kind of platform. So now this figure here illustrates a workflow that could be utilized for the planning and implementation of multi-omic clinical studies. When considering study design, selection of biological material for common omic analysis, data integration, and reporting of findings to patients. So, and there is a need for discussion on ensuring the data we generate is publicly available, whilst protecting patient confidentiality to enable large-scale collaborative efforts. Now, a phenomenon which would be particularly helpful for diagnosing currently undiagnosed patients. Such a discussion and development of resources should involve continuous consultation with patients and their family members. We can start by the study design. So how to design a study design. So how to conduct power calculation to estimate required sample sizes. So where possible, collaborate globally to maximize the recruitment pool. When recruiting, record phenotypic data as much detail as possible. And then the next step, you go for sample selection. And now you can see, you can map it out. And as example, in this table, the sample type. What would be the sample types in medicine? You can get uh, hair follicles or dental plaques, saliva, buccal cells, plasma serum, particles, sweat, urine, feces, semen, biopsy samples, and which common omic analysis are possible. So for example, hair follicles, it's possible to do epigenomics, genomics, proteomics, metabolomics, transcriptomics. But how about dental plaques? So far, well, genomics, yeah. We don't have microbiome here, but dental plaques is accessible for microbiome. For plasma or serum, it's possible to run a proteomics or metabolomics, but it's not, it's not suitable for analyzing transcriptomics and so on. So, and then the individual wet lab protocol. So once you establish this, and then what would be the wet lab techniques that you're going to use, whether it's microarray or next generation sequencing or mass spectrometry. And each of those techniques will generate data and needs to be analyzed. So, and how to integrate between the first omic, the second, and the third omic. So how to run an early integration, concatenation of multi-omics data into a single matrix. For example, one of the platform software uses deep learning called autoencoder, or late integrations. When you analyze each of those data sets and clustered for a similar features, and the cluster are integrated. So for example, similarity network fusions. I think you would remember the previous slides. And obviously, what is needed is statistical integration. So we want to see probabilistic inferences about multi-omic. So for example, this software called Paradigm, and how the statistical integration would work. So, and then it's getting more challenging, but because you need to do a replication. So like Koch principles, you need other than replication, we want to see reproducibility of the data generated by different kind of omics. And then obviously you need to validate this data. So whether you want to use Sanger sequencing, validation of next generation sequencing, or just follow the Koch principle, or you will see how an example we'll discuss later, they validate their discovery. And obviously you need to discuss with multidisciplinary team what are these findings, meaning what are the findings means to the patients, to the scientist itself. So, and in the case of medical study, it needs researcher, the scientists themselves, clinicians, and genetic counselors, because it involves DNA data of patients, and how you want to write it into a report and to support. So anyway, so where I've been discussing theoretical part. I know it's challenging to listen or to read the theoretical concepts of multi-omics. So why not we discuss an application of multi-omics that the discovery led to a potential nutritional amendment to overcome a health issue. So this is a public case study entitled Longitudinal Multi-omics Reveal Subset-Specific Mechanisms Underlying Irritable Bowel Syndrome. Notice here, you've got people with various expertise here, such as from the top of the list: gastroenterology, hepatology, metabolism, digestion, reproduction, biological sciences, computer science, engineering, genetics, cell biology, development, health sciences, system biology, applied mathematics, microbiology or immunology, physiology, biomedical engineering, cancer, microbiome. So it is truly a multidisciplinary effort to apply multi-omics and unravel their data. So let us briefly hear what their summary is. So I'm going to activate here, and let me know if you cannot hear, but I share the sound. Hang on. So
Irritable bowel syndrome, or IBS, is a globally prevalent disorder predominantly seen in
I hope
Irritable bowel syndrome,
I
or
hope you can
IBS,
all hear
is a
the.
globally prevalent disorder predominantly seen in females and is characterized by abdominal pain or discomfort, combined with changes in stool form or frequency. Based on the predominant stool form, IBS is divided into constipation predominant, diarrhea predominant, or mixed subtypes. The gut microbiome, the large collection of intestinal microbes, genes, and their products, has been implicated in IBS. As a result, patients often use probiotics to treat IBS symptoms, but they are not very effective. This is partly because multiple factors are involved in IBS, and we do not understand how the gut microbiome fits in the larger picture of IBS pathogenesis. In this study, we collected multiple layers of data over a 6-month period, including diet, patient symptoms, measurement of intestinal secretion, gene expression changes in the GI tract, gut microbiota composition, and the compounds present in fecal samples that can originate from the microbiota, diet, or the host. We integrated all this data into a meaningful format to unravel biological relationships in the highly dynamic ecosystem of the gut. By doing this, we discovered a new pathway that is important in IBS. This involves the metabolite hypoxanthine, which is important to keep the intestinal barrier healthy. We found that altered hypoxanthine metabolism involves both the host and the gut bacteria, which highlights the importance of these type of comprehensive studies in humans. We also identified additional bacterial compounds that may be important in IBS. For example, in patients with diarrhea, there was increased bacterial production of tryptamine, which increases water secretion in the intestines. These patients also seem to harbor reduced bacterial metabolism of bile acids. As a result, high levels of primary bile acids were present that can irritate the colon and increase water secretion. In patients with constipation, production of short-chain fatty acids was decreased. These bacterial compounds normally increase movement of the stool across the intestine. Based on our findings, native or engineered bacteria that produce short-chain fatty acids or tryptamine may be used to treat IBS with constipation. At the same time, bacteria that convert primary to secondary bile acids may be used to treat IBS with diarrhea. Finally, hypoxanthine-producing bacteria may help treat symptoms in patients with both subtypes of IBS. One of the strengths of collecting samples over a 6-month period is that even when following few patients, gut microbiome changes can be identified that precede worsening of symptoms. Interestingly, these changes were not necessarily consistent between people, which explains why the same treatment may not be suitable for everyone. Overall, our study highlights the importance of longitudinal sampling and integration of multi-omics data with patient data and GI function to identify novel treatment targets. Irritable bowel.
So I'm going to elaborate quite a bit from their study. So the rationale to conduct such a clinical study was as follows. The human studies in IBS are limited by cross-sectional sampling and lack of subtype stratification, which is reflected in the lack of agreement in finding across a large number of microbiome studies. So the latter further increases variability given the well-described influence of gastrointestinal transit on the gut microbiome. In addition, IBS, like other chronic gastrointestinal disorder, is characterized by periods of remission and exacerbation in symptoms. And cross-sectional samples thus fail to account for temporal variability in disease. So now, how do how to study, how to design the study to address the issue. So this is what we want to learn, how designing study of this, how to they strategize the experiment, and take a look here the number of sample for each omics approaches, the dietary survey. So they would note each diets of each patient. There, the N is 433. And metabolomics, there are three methods with, so the N would be 1220. And metagenomic, the N, the sample size is 474. So, and this is once a month, and the stool, total stool analyzed was 474, right? You can deduce here all the stools undergo metagenomics, so 474, and not all would yield data for dietary, but metabolomics, they run three different methods, and 1220 divided by 474. Now you get roughly the number, the data, the sample size for each methods. And the biopsies done before and after the 6 months. So, and from the biopsies, they run 16S rRNA analysis. There are 72 samples, and metabolomics using GC&MS, there are 74, transcriptome 68, methylome 70, physiology 72, cytokine 63. And from the serum, they also run metabolomics, this time using LCMS, 78, and cytokines 60. So the sample size itself, you can imagine there are differences in sample size, and differences sample size would reflect differences in the host, in the phenotype. So now you can see, how do we harmonize this data? That's the challenges. So, but we want to understand a little bit more by understanding a range of methods employed to address the issue. While the methods are categorized, the question for you to ask is, how do they determine what kind of methods are selected, which methods are executed first, second, and so on? Or how do they determine the execution sequence of the selected methods? So let's take a look quickly the methodology they employ in this clinical study, and you notice how thorough they execute the clinical study. So first, the recruitment and exclusion criteria. So how do they recruit patients? They advertise it using newspaper, flyers, letters, or word of mouth, or from websites, and the range of subject, healthy subject, and patients 18 to 65 years old. And the two sigmoidoscopy would need to meet Rome 3 criteria of the clinical exclusion inclusion criteria. A healthy subject may match patients for age, sex, and BMI. So now we understand that we are dealing with nutrition. BMI is an important part. And exclusion criteria, it's important to be established. So this is, for example, prior abdominal surgeries. If they had the IBD before, colitis, celiac disease, antibiotic use, bleeding, or pregnancy, or plan to be pregnant, or less than 18 years old, older than 65 years old, others as judged to non-compliant or adversely affect the outcome. Now, the informed consent, the regulatory compliance is a must, and so they have to establish that. And how the participants and the sample metadata. So the first visits, they would note all the medical history, limited exams such as they would note height, weight, BMI, vital signs, and then undergo dietitian consult. They would run FFQ and 24-hour DRQ, and based on the Rome criteria to determine IBS symptom severity, and so on. So they would note quite a detailed medical history during the study. And animal experiments. The question, why do they need animal experiments? Will we're gonna touch base on that. This is important to validate their discovery. And of course, the specimen collection and data generation, the stool specimen, they were provided by home collection kits for the first one, and monthly they would drop it off or FedEx it, send it by post in ice pack, store it in -80 degree. And blood samples on the first visit only, and then be stored in -80 degrees Celsius. And biopsies, like sigmoidoscopy. This is the area where they would take the biopsies, sigmoid colon 20 to 30 centimeter from anal verge, and save it in RNA later solution to prevent RNA degradation, or liquid nitrogen or glucose scrap solution. Now, hang on. It's quite a lot using chamber experiments. I'm going to go quite quick on this. And then the microbiome DNA sequencing. Obviously, you need a DNA extraction from stool biopsies, and then a set of an array of wet lab work from quantification, run the shotgun metagenomic, and then bioinformatic work, alignment to reference sequence, prokaryotic genome, and then functional profiling using KEC orthology, and predicted profile augmentation, and then alignment. And the microbiome data analysis itself, the taxa, and so on. Then we have a metabolomics using what is an NMR, serum and stool sample, and subjected on a Bruker biospin 600 megahertz, then analyzed using MATLAB, check the spectral data. And then they also run LC-MS-MS or GC-MS-MS, depending on the samples, and then they transform the statistical significance in R in several packages. And they wanted to find out the cytokine measurement using Luminex Bioplex 200 to measure various interleukins, interferons, TNF, PGF, leptins, MIG. And another big data generating would be techniques would be RNA sequencing analysis from the biopsies. They isolated the RNA and then run it using intrinsic RNA library prep, and then check the gene expression count using several packages over there, MAP-R-seq with top hat, then annotate the gene using ensemble, their RPKM, and then check the differential expression, and they use XR and the pathway enrichment to find out, and using R packets right-hand. Then how about methylome, the epigenomics data? So they run from the biopsies, they run 850K CpG Illumina methylation beats, and analyze the data using R package and check the differential methylated CpG. And finally, the integration. So how to integrate all this big data? So for example, stool microbiome versus metabolites. So they use different packets, what they call it, Maslin-2 packets in R with FDR 0.25, and all taxa abundance data, CLR transformation, correlation species network, and LASSO to understand gene microbiome gene metabolite association used in our packets. LASSO means this is a statistical analysis. It's least absolute shrinkage and selection operator, LASSO. So lasso regression is a type of linear regression that uses shrinkage, or shrinkage meaning where data values are strung towards a central point, like the mean of the data. So the lasso procedure encourages simple, sparse models, models with fewer parameters. And this particular type of regression is well suited for models showing high levels of multicollinearity. or when you want to automate certain parts of model selection, like variable selection or parameter elimination. So, and yeah, so there are quite the data, and well, we still have in vitro and in vivo hypoxanthine consumption experiment. Now they have a discovery, and then they have to develop a bioassays, and they run in vitro and in vivo. So they would culture the bacteria, and then they check the hypoxanthine using LCMS, and they monocolonize the 6-week-old germ-free Swiss Webster mice with three mice with 2 million colony forming unit. So, and yeah, and then they check the cecal contents stool of the mice using this assay, amplex red xanthine oxidase assay kit, and then quantification of statistical analysis. So you would be amazed on this study was done thoroughly, and they uphold the principle of Koch postulate. So, and we've seen this, the methodology, the case study, and you can you've heard the description of the outline of the sampling, and then they come up with hypoxanthine that is significant as the metabolite, the hypoxanthine in the stool, and then they ran the monocolonization in 3 Webster's mice cohorts, and then they have a follow-up longitudinal study. So you gather the bacteria into the mice, and add hypoxanthine, 100 micromole hypoxanthine in drinking water, and then on day 4 they collect cecal content and then check. So they culture it using Bifidobacterium longum and Lachnospiraceae. Yeah, and then they measure the cecal content of hypoxanthine. So now they analyze the data here. So, and they use an enzyme assay. Sample were corrected for baseline level in hydrogen peroxidase in the sample based on a parallel reaction without xanthine oxidase enzymes. And yep, so, and they have a data plotted in here. All right, so yeah, it's something that I like to repeat what their conclusion and is. I like to quote them again what their conclusion is. So their study provides multiple new therapeutics targets for future investigation. So the apparent decreased microbial biotransformation of BA in IBSD patients might be treated using defined microbial consortia with a high capacity of conversion of CBC to LCA. So similarly, in IBSC patient, increased production of bacterial SCFAs and or tryptamine may be viable therapeutic strategies. Finally, stimulating microbial hypoxanthine production or inhibition of xanthine oxidase locally in the gut would be a novel approach to increase the amount of luminal hypoxanthine without systemic effects, and may be beneficial in IBS independent of disease subtypes. That's their conclusion. They have a discovery. They've done thorough study and massive data. They try to bridge a platform between with different kind of omics. And however, at the end, the wonderful things these scientists do is to admit their limitation. This is a good example for all of us. Here what they say. The limitation, quote, we acknowledge that our study has some limitations. we realized that our findings are not proof of causation, even though we primarily focus on biologically plausible mechanisms. In order to increase rigor and confidence in our findings, we integrated multiple data types and primarily reported pathways that were identified across different data types. We focus primarily on the colonic microbiome, but we are aware that the small intestine likely plays an important role in the generation of IBS symptoms. Longitudinal studies dedicated to the small intestinal microbiome are needed to complement our findings and advance our understanding of IBS. In addition, longitudinal multi-omics data presented in this study could help in moving away from phenotype-based to mechanism-based IBS subtyping. So I'm going to stop the presentation, and if you guys have questions, feel free to ask. So we have quite a bit of time to have a discussion. So this is an introduction, and there is a literally there. It's really good examples of a study that you aim to collaborate between different kind of fields, and this is a good example to run collaborative works and where different kind of expertise put in play, and we can have this multiple interdisciplinary technologies and data, and now it's possible to use the bioinformatics. And if there isn't any available that would match to the study, so the thinking is the other way around. So what would be your study? So the bioinformatics would follow, and based on the study, then you see, oh, there isn't available in the market or in the platform of really available softwares or platform. Then you would need a programmer to help to build the multi-platform or the integration between different omics in your case. So anyway, so I'm opening up for questions.
Yes, thank you, Dr. Felix, for the presentation. And if the participants have any question or maybe have some thoughts about the presentation or have some experience related to the topic presented by Dr. Felix, maybe the participants can share it or can ask through the chat feature in Zoom or can ask directly. Thank you.
Any questions? And anyone here have done not multi-omics, at least single omics data analysis?
Yeah, we still have quite a bit of time before we end the session, so please use this time to discuss the topics. If there are any questions or maybe experience related to the topics, maybe the participant can share it and discuss with Dr. Felix. Yeah, Dr. Mifa.
Thank you.
Yeah.
So I want to ask a question. I currently joined the research that uh, correlating the gut inflammation and stunting. Uh, the idea is that when the gut condition is bad, then there will be like inflammation there, and the absorption of nutrient will be bad, and it will affect the growth of the children. But I also read in other research, and the method of to measure the inflammation is using ELISA, measuring the IgA, secretory IgA in the stool, and then but I read in other research that apparently the IgA concentration most likely didn't correlate with the stunting. So I want to ask, is there any method that more sensitive, or yeah, to do that? Thank you.
Yeah. Okay, thank you, Mifa, for the questions. So, so you want to correlate the now you can have a new term here, not really a new term. You want to correlate in inflammations, or literally you want to study inflammasome. What is inflammasomic? So that would be correlated with stunting. So the inflammasomes literally cytosolic multiproteins, oligomers of the innate immune system, right? Responsible for the activation of inflammatory responses. So it means you need to identify who's the player in here. So, but if you were to just use IgA, obviously it's nothing specific because this is only a type of immunoglobulin. So, but if you were to use Omics technology, then one of them is using the in what we call inflammasome. So there are quite a players over there. So different kind of players. So caspases, for example, in the caspase pathway. So the NF kappa B activation, and if you learn some of the cellular signaling, it's quite crowded traffic over there. So and that's the theoretical part, but learning from today's study and examples in the lectures, they're using what we call hang on, I'm going to open it up again. They're using in their methodology the cytokine measurement. Cytokine is one of the one part of the, it should be one of the part of the inflammation. However, if you're to look the Luminex product, Bioplex 200s, they list various interleukins, interferons, tumor necrosis factor, vascular endothelial growth factor, leptin, and then that's part of the inflammasome. So, and you can correlate this. So they have this Luminex Bioplex 200 system. They have 100 analytes in single microplate. So it's quite a data there, quite a numbers of cytokines and so on. So but this is going to be like still a single omics, right? So if you were just to focus on the immune system, any factors that creating inflammations, that's going to be a single omics. and you correlate it. Of course, statistically, you need to correlate it with the criteria. What would be your criteria of stunting, and what would be the exclusion inclusion criteria, as usual, the clinical criteria. Now the question is whether you want to correlate this inflammasome with the gut microbiome. So now you move to the second omics there. So now, so what happens to this patient that meet the criteria of stunting, and then you want to find out what would be the microbiome dynamics in the gut system. So, and as you learn from today's lecture, the study that was presented here, they argue a longitudinal study would be better off to understand the interaction between this omics. So if you just capture at one time, one point, cross-sectional is unlikely to yield meaningful data for this patient. So a longitudinal study would be a perfect fit. So for example, when to capture the inflammation, if you were to, if the patient were to be biopsied and in what region, so you need to determine, right? They admit that their limitation is only the biopsy is only one part, and they notice the intestine, the small intestine likely play a factor. So that's should be a debate, debated discussion, or argumentation has to be established. How are you going to sample this before you run it into an omics data or wet lab experimentations? So that's a critical point. Now you have two, right? Inflammasomes and microbiome. How about the genetic factors of this person? Because you want to correlate again, or you want to exclude, there is no genetic factor, it's purely environmental. In this case, the input of the nutrients. So the genetic data would be genomics or epigenomics, no, the third and the fourth omics. How about environmental factor, the nutritional itself, nutriomics, nutrigenomics. So, and you measure it by checking the metabolomics, by using metabolomics from the diet. So now you can see how the multi-omics play a role in this case. Just from one study, the topic is stunting, then you can expand by understanding how this scientists, put together all the design, and we can immediately expand it into like a comprehensive. Now the critical part, how to integrate this data, right? So that's what we learned today as the introduction to this multi-omics and for this nutritional applications. And I hope it helps you to answer the questions. Surely IgA only one answer. So we have different types of IgA and they won't be specific, but if you were to use multi-omics, there is this inflammasomes to study. There's also microbiome if you want to understand the dynamics of the gut microbiota in the patients, and metabolomics from the nutritions. Okay.
Yeah.
We have a second question here on the chat.
Yeah, from Miss Randy.
The background is agricultural molecular analysis of fusariums, the aflatoxin and humonicin and its effect of human body. Would that be possible to run a multi-omic analysis? Yeah, let's expand, right? So obviously you cannot study it right away to human to perform clinical study of this aflatoxin of humonisins, right? So, but a model of this aflatoxicity in, or if you were to develop an animal model of aflatoxicity or fumonisin induced toxicity in an animal model, in small animal model, to get an understanding, then you can also expand the study by using multi-omics analysis. So obviously you're not going to use fusarium assays because it's complex. So even though you try to come up with one particular pathogenic strain, but the conclusion is almost never to generalize the fusarium toxicity. So right, so the good to establish is to one way to approach it is to use a pure aflatoxin or fumonisin in this case, or both, right? So then you get a dose dependent range and what happens to the body. So you establish toxicity and what happens um, in the liver functions or in different kind of metabolisms. What is the pathological outcome in the small animal study? So you have a histology or a histopathology can be established, and if you were to grab the biodistribution, I study biodistribution. I use PET CT scan, and obviously not just the PET and CT, but literally the subject, in this case, the small animal model, would be sacrificed. And then you grab all different kind of organs, from brain to you name it, different kind of organ. We see the biodistribution. So the small compound and the targeted, and then plotted what happens. This is by itself is kind of like an omics, because you generate different kind of images. So of histopathology. So that's one aspect. So now, if you were to trying to understand the overall what happens, let's say if the target, the immediate target, the liver toxicity, you want to understand how the liver toxicity of this aflatoxin or fumonisin in this small animal model, then you can harvest the liver, and then you check the transcription, the transcriptomic, the alteration, the genetic alteration, the genetic expression alterations during the course, and then you can correlate this with the dose dependent, depending on the dose, the exposure of this toxin, and that's another aspect to tie it up. And then you now the second omics, right? Then you can tie it up with the histopathology, try to correlate. I know this is a picture and this is the data. How to integrate this? So that's the challenges in the multi-omics study. So once you establish it, now you want to see whether this data would be reproducible if you were to see it in human. So, and if you were to approach clinical study or clinicians, medical doctor or scientists specializing in mycology, clinical mycology, and then they would be able to help you to narrow it down. Okay, this phenotypic, this patient with this kind of criteria has been diagnosed, potentially have a certain exposure against this fusarium or aflatoxin, and based on numbers of blood works or data from blood works or different, or even biopsies. Now you can try to match this data, and basically you can check by using, let's say, a serum, and then check the metabolomes over there, or proteomes, or even transcriptome, and then try to see if there is a correlation with your small animal model with the clinical outcome. So now you try to correlate this with clinical study. Then you need another integration in the bioinformatic levels. So, well, this is just an example, like instantly trying to expand one particular simple biological question to have a layer of studies and how to integrate it. So, and okay, I hope it helps to answer your questions.
Dr. Felix, there is another question in the chat feature from Mrs. Rizka Nindya.
Yeah, my question from your experience, what is the most challenging part when you want to start research in this area? What would be the most challenging part when you want to start research? The research question itself sometimes is quite easy, but if you learn to the study design, so my experience as a reviewer, reading different kind of submitted article in genomics, the biggest issue that I faced was actually in the study design. They're lacking of reproducibility, and it was basically not even multi-omics. Even one single omic, that's a transcriptomic. So I'm going to give you an example. So the transcriptomic, it means you are trying to capture genetic expression at the level of RNA. The question is when to capture it. So the same question that was when these scientists, the publications dealing with IBS-C, IBS-D, they determine, okay, this is the regions to biopsies, to take the samples and then to preserve. So it's always we have to establish an argument when to capture it, and how do you control that? Because you are dealing with massive data, and slight differences would completely change the landscape, and hence you need replications. So that's critical. That's critical, and why it's going to be a challenging, because the minute you start to make a decision to embark on this path, it means it is still expensive study. So, and the technique itself, all the tools and equipment and the running its data, it's quite expensive. And so it means not just how to capture, how to replicate, how to control it. And if you let it, if you have the freedom, then you can see it's going to add quickly, the number of samples and how to control this, how to, and then the criteria to focus. So imagine dealing with the IBS-D or IBS-C, they come up with different kind of metabolites, and they know there are differences, but why they pick xanthine pathway, they come up with, you've got to make a decision for certain pathway, and yet you need to validate it, and you have to follow the Koch principles in this case. So that's getting it quite complex, and in the level of difficulties, obviously the computation is for those in biomedical sciences, in medicine, definitely is a challenge. So you need to outsource to those who are proficient in their own field, understand how to code, how to create a program. However, what to code, what to program, how to link, that becomes the principal scientists that would need to strategize this. I want to have this correlation between pick up the the case of inflammasomes and stunting. So stunting would involve not the anatomical part, the nutritional condition, the BMI, and so on. And then we want to get this data into the system and then to correlate it. If we don't have any available platform out there to take the advantage of, then we need to create an in-house program. And so you've got to be proficient in Python and in R to trying to get what part of statistical analysis would be deemed suitable to run this analysis of the multi-omics. So, well, at least what I can say today, you've learned from something very simple questions, and I'm grateful for questions as simple as correlating stunting and inflammations, gut inflammations, or as simple as the question of aflatoxin or fumonisin from Fusarium species and how to expand it. The same thing with IBS. Oh, we have an IBS in the patient, mostly female, and then we categorize IBS-C, IBS-D. Okay, the control is healthy, and then they have to match and everything. Then expand it significantly. So now the architects behind this is the one that would have would need. I must say, he or she would his or her task would be one of the most challenging. Strategize different fields. Technicality, you can collaborate with the expert in the field, right? But I hope it helps you. So start with something simple. In life sciences, there are numbers of biological phenomena that life sciences is really difficult. As far as I studied life sciences since early 90s until now, honestly, it's really difficult. I don't want to compare with mathematics or physics or chemistry, but life sciences, you are not dealing with one size fits all or 100 is 100. No, you always have intervals. You always have different layers, like today's example, and how to integrate this. The mindset cannot be linear. So like in mathematics, it's really exact. It's nice, and you can do a linear concept. That helps because human inherently, it's easily to grasp something linear. Something linear, meaning if you have a dimmable lights, and you can dim from 5 watts to 10 watts or 50 watts, 100 watts, from dim to bright to brights to really glaringly bright, for example. But in life sciences, this is how to think to strategize a study using different layers. So today it is a good example for you start to understand life sciences from different aspects. So, and at the end, the conclusion, we never say, okay, this is 100%. No, there is this particular situation, and this is the likelihood, and it might be confounded by this and that. So there are boundaries of the study that can achieve, and it's subject to correction. So by larger study or by a different demographically different study, the same study but a different demographic, for example. So okay, I hope it helps you to provide a perspective on this. It's another. Yeah, found some difficulties to understand, and in the chat there is especially molecular analysis. I haven't seen much research in Indonesia analyzing the mycotoxin in human body, for example. Gut microbiome in our intestine. Trying to find a PhD position on this topic, but I haven't found one. Yeah, I hope it helps you to get a perspective. And when you create a simple biology question, of course, you need to check the literature, investigate and consult all the literature available in the world, and then see whether someone else has an approach to answer that question. But what kind of angle or needs that hasn't been discussed, or at least you understand aware what is the current situations with that particular subject or topic or study. What is the big question? What is the or nagging questions in that fields, and try to get the angle. So when you run, especially an advanced study in the PhD level, you need to get down into the molecular levels to understand mechanistically. And in life sciences and medicine, validation is critical, and not just validation per se, but the Koch principle. Remember the Koch principle. So that is critical. And so I forgot to mention one thing, because the biological phenomena, because we in a population study, the mindset of Darwinian thinking, it's also clearly important. In the population, when you suppress one subset of population, there is going to be an effect. There are numbers of other subpopulation that is thriving, or vice versa, or there is an kind of a tug of war, different subset or different populations. So that has to be in the mindset. So called Darwinian thinking of the population is also critical to be part of a consideration when we develop the study. So we still have some times. So feel free to have any other question. Maybe I speak a bit too fast, so it ran quite quickly apparently, the prepared presentation. Any other questions?
Any other question from the participants? Yeah, is there any other question from the participants? Oh, Dr. Mifa would like to ask.
Yeah.
Yeah.
Continuing to my first question about the inflammation biomarker. So in our research, other than the IgA, we also measure another cytokine like interleukin, interferon, and the other inflammation biomarker. And also we measure the methylation profile of the subject, also the microbiome profile of the subject. And then I know you already explained how to to do the the omics analysis, but can you give me the recommendation, like more specific about our research?
Okay. With regard to, yeah, as you said, you mentioned some of the specific cytokines or whatever inflammatory agents, or we are not talking about cells in this case. So yeah, that's a correlation. But with there is a questions that I might ask is for the epigenetics study, the epigenetic study, or if you were to use epigenomic, the totality of epigenetic modification or methylations in the I would assume in the guts. Well, it can do methylation can happen in the whole cell, so in the gut cells, or as well as in the bacterial cells, in the microbial cells. So that's something to be discussed quite deeply. What would be the rationale? So rationale, the reason to use particular techniques, that's something that you need to take a time to debate, to argue why you are using epigenetics. And if you were not to use omics, if you were to just study, let's say, cell signaling, molecular biology, we would go down into the molecular levels itself. Why this particular molecule be a target? What would be the pathway look like? How do we identify the communication between this in the interplay, the communication between these molecules? It means the level of phosphorylation. It means you need to run immunoprecipitation study, for example. But in the level of epigenetics, why and how it would play a role in the stunting. Your patient, you have already decided, okay, the phenotypes that you're going to study is the stunting, for example, and there are two immediate. So when we simplify in life sciences, there are tug of wars between the genetics, the factors, the inheritance factors, and the environment. So in the case of stunting, we could investigate the genetic factors, we could investigate the environmental factors, we could investigate the nutrient, intakes over time, what is missing, what is the issues, either this issue, and obviously there are numbers of environmental factor quite wide, then you would need to narrow it down. So the microbiomes, would you consider microbiome genetics would affect the genetics or the environmental? So the genetic factor or the environmental factor? We might tend to say, okay, this is an environmental factor. So I can still see the link of the microbiome. So and something to remember is the microbial composition in the gut depends heavily from the nutrient intakes. That would be the immediate. So you change the diets, you would change the dynamics significantly. However, it is correct that you want to study the inflammasomes, because this would determine who can survive, what subset of population of microbiome would survive, and how this subset of population, or the survival of subset of population would contribute to the phenotypic, the stunting phenotypic. And see, there are numbers of questions to ponder, to discuss, and then you that probably could answer that. It's going to be challenging which one of which before we go to the technical. So in the study of life science or medicine, I would suggest you try to start with the clinical case, with the biological phenotype you want or phenomena you want to study, and then worry about the technical later. So once you establish this, the path, and then you start to look which methodologies available, which are not, how to develop. So if you don't have an available methodology to answer that question, then you need to develop the bioassays to bridge this gap, right? So yeah, I hope it would help you to ponder, to answer, and obviously you need to discuss with different I don't know, colleagues, supervisors, and then to pin down what exactly you are looking for, and then we can easily uh, 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 find or point it out. Okay, this methods would be available. So some methodology you won't have any choices. So for example, sequencing, the genomics, or the transcriptomics. So I think predominantly it's going to be the Illumina NGS techniques, right? So it's hardly you have an alternative. There is a nanopore, but it won't be as, the data won't be as that extensive as those Illumina. So no, the output from the Illumina and nanopore, if you were to use two different methods of the sequencing, yet you need to have a statistical correlation among them. So sometimes you don't have quite an option. Let's say the available platform would be the Illumina for sequencing, or the discussion on the IBS for metabolomics. They use three different kind of methodologies: the NMR, the GCMS, LCG, MSMS, and GCMS, and yet the input to run the analysis from different samples, and you would wonder why it's available only to these particular samples, although you are using metabolomics. So I think as far as the information of for the stunting, that's so far that I can provide the guidelines. To go into the detail, obviously you need to come up with a path of the study. Then the technique can be suggested with techniques. So, and speaking of the bioinformatics, it's going to be later, way later. the bioinformatics analysis, so it's going to be way later. Yeah, there are numbers of options, but it's pretty much still in the designs level, right?
Thank you for the insight.
Oh, I hope it's answer your questions. At least provide you some perspective.
It's good. I think I should like find the link between the.
Okay. Okay. Yeah.
And we have around 4 minutes before we end the session. If there are no other question, I would like to take pictures first with all of the participants. So please turn on the camera if possible.
There's a questions whether you can get the PPT. I will have.
Oh,
I will send it to the doctorate program
okay.
at Faculty of Medicine, so we can have access. You can have access to the PowerPoint presentations.
Okay, thank you, Prof. Alex.
Yeah.
Yeah. So please turn on your camera. 1, 2, 3. Yeah. Okay. Thank you, Dr. Felix Mesak, for the presentation. It is very interesting and very insightful, especially for all of the participants. And I would like also to give my gratitude to all of the participants who have been attending this event and very active in the discussion. And so I, representing the webinar committee, apologize for any mistakes and shortcoming, especially from me. And thank you very much for your attendance, and see you on the next event. And the PowerPoint, maybe I will send it via email to the participants. Okay. Thank you. So please.
Thank you. Have a nice day.
Thank you,
Bye.
Dr. Felix.
Yeah,
Good night.
Thank you.
welcome.